Good morning, everyone. Today I am going to explain you the different stages of mitosis and cell division. As we know that life starts its journey from the single cell zygote when the sperm and ovum meet together, and then multicellular pattern of embryogenesis, and then multicellular body, millions of cells are being added when time passes. So there are two main type of cell divisions. One is mitosis, another is meiosis. Students generally ask this problem. Try to understand how can you define and what are there. Okay, so first you know what the mitosis is. You can easily define. Nothing I have remembered. Okay, just this chart is in my mind and I am defining this. Try to understand. This is mitosis. How can you define this mitosis? It is a type of cell division, write down. It is a type of cell division in which one mother cell, this one is the mother cell, the cell which divides is called mother cell, and the cells which are formed are called daughter cells. So the cell divides mother cells. So one mother cell divides once to form two daughter cells having same number of chromosomes. When female is there, then both of them have equal number of same number of chromosome present, then in comparison to the mother cell, same number of chromosome to the daughter cell. So type of cell division in which one mother cell divides to form two daughter cells having same number of chromosome is called mitosis. And what the meiosis is? The meiosis is that type of cell division in which one mother cell divides twice, once and second time, twice to form altogether four daughter cells having half. It has 2n and it has n. So half number of chromosome as compared to the mother cell. Remember, meiosis occurs only once in the year. In the whole life, meiosis taking place once to restore the diploidy condition, to make the gametes diploid so that they unite further to give rise to zygote, which is diploid, and to form the body diploid. So diploid to diploid to maintain the constant number of chromosomes. Meiosis is evolved in sexual mode of reproduction. So what happens in mitosis? They are further divided into two stages. One is the karyokinesis, another is the cytokinesis. Karyokinesis means division of karyon means nucleus, and cytokinesis is the division of cytoplasm. So what happens in karyokinesis first? First karyokinesis taking place, then cytokinesis. It doesn't that cytokinesis taking place first and karyokinesis later. No. Karyokinesis means division of nucleus will be prior to the formation of cytokinesis. Kinesis means breaking down. Whenever in biological world, kinesis means or lysis means breaking down, synthesis means formation. So here in prophage, it is further divided. Karyokinesis has different stages like prophage, metaphage, anaphage, telophage. Four phases are there. These all are characterized by a peculiar changes taking place in the nucleus. Karyon means nucleus. So what happens in prophage? It is the longest one first, and chromatids, which has no identity. Chromosomes has identity. You can visualize chromosomes. Chromatids are thin, long, slender, uneven, so you cannot see them. So chromatids, which are unseen, indistinct, now become distinct and they become shorter and thicker and take the identity distinction. It's distinct. You can easily recognize them. Centriole divides, chromosomes are shortening, thickening. Centrioles divides and start moving towards two opposite pole in the early prophage, and late prophage already moved. The chromosome become condensed totally and come to the equatorial plane. So these are the two diagrams showing the early phase, early prophage. This one is the early one and this one is the late one. So here the nuclear membrane, nucleolus, cytoplasmic reticulum, Golgi bodies are appearing to be disappear, and here they are totally disappearing. So in this way they are distinct. So then prophage passes away. Then come to the metaphage. So what happens in metaphage? Metaphase is characterized by the chromosomes comes on the equatorial plane. Try to understand. This is the difference between mitosis and meiosis. Chromosomes attach to the two opposite poles. The spindle elongates from two opposite poles, and each chromosome attaches with these two spindles. Means suppose that I am chromosome, I have been attached from both the sides. When they elongate, the spindle come from both the sides and attaches to me. And then they contract. Then they split me into two equal halves. But this doesn't happen in meiosis. In meiosis, only one chromosome attaches to one side. From this side, no. So when they condense, when they contract, the chromosomes, the whole entire chromosome move towards one pole, not the half. So chromosome number reduced to half there. What we have seen in meiosis first. So here condensation of chromosomes is completed. No nuclear membrane, nuclear body, endoplasmic nucleus are seen. And spindle elongates from the centrioles and finally attached to the kinetochore. Here kinetochore is the part of centromere where the spindle attaches and chromosomes are arranged on the equatorial. No nuclear membrane, nucleus and endoplasmic reticulum are seen here. The metaphase passes. That's one. Anaphase. What happens in anaphase? In anaphase, it's a very fast stage. Immediately it passes. What happens? The contraction begins in the spindle. So due to contraction in the spindle, the chromosomes split and the chromatids. So very fast process, contraction in the spinal fibers, chromosomes split through the centromere to form two daughter chromatids. Now they are called chromosome because they have already duplicated in S phase of the interphase. The daughter chromatids move towards the opposite poles, giving L, V or I shape depending upon the morphology of chromosome you have established. So that their arms they are moving in such a way that their arms are directed towards the center and poles, centromeres are directed towards the poles. Suppose that this one is the chromosome, and when it moves in this way, so its centromere will moving toward this one and arms remains there. So it's a thread like simple. When you pull the thread, it turns like this way. So you can see here L, V or morphology of chromosome can easily be seen. Next is the anaphase passage. Telophase. Nothing extra in telophase. It is reversal of the prophase. So what happens in prophase? The reverse will happen in telophase. Now nearly the chromosome has divided, has split, an equal number of chromosome has given to both the poles. They have organized. Now they require nuclear membrane, nucleus, so that two nuclei are to be laid down to be formed. So in this way, here the reversal of the prophase, the chromosomes have reached at the pole. Chromosomes have already reached at the pole, and reappearance of nuclear membrane, nucleus, Golgi taking place. These all are nuclear membrane, nucleus, Golgi there. All are cell organelles are to be reappeared. What was disappear in prophase? Hence it is a reversal. Chromosomes become thin. They lost their identity and turn into chromatids, and it leads to the cytokinesis. After that, this is the final stage of karyokinesis and leads to the cytokinesis. So what happens in cytokinesis? Cytokinesis is defined as division of cytoplasm. It is differ in plants and animals. The questions generally ask that how, in which way it is different in plants and animals. Okay. So in plants, okay, so this is the diagram of plants. The rectangular cell I have made. So it starts from the center itself and moves towards the periphery. In plant, cell plate formation begins from the center and moves slightly towards terminal part. But in animal cell, the fissure develops from the terminal part and deepens gradually and split the two one mother cell into two daughter cell. So in this way it differ from the cytokinesis differ from the in plants and animals. In animals, the whole develops from the margin and deepens gradually towards the center. Develop from the margin and deepens in this way. Develop from the margin deepens towards the center. But in case of plant cell, the cell plates are formed from the center and elongates towards the periphery or the margin. So in this way, two daughter nuclei are formed and each have nucleolus, nuclear membrane, cytoplasm has already been divided. So one mother cell is totally divided into two daughter cell, and in this way, asexual organism, simple organism, unicellular organism, they reproduce by the means of mitosis simply. So two daughter individuals are produced, and our cells in somatic type of cell we generally divide to add the similar type of cell. Okay, thank you.